All right, that was great. That was like really awesome. Okay, guys, so I gotta go lecture. I'll see you guys after. All right, hey everyone, how's it going? Hope you had a fantastic first week. We're into a second week and we're actually into a slightly different module. It's not that you have to forget everything that you've learned so far. I need you to actually make sure that you're staying on top of the material because it's building on each other. Now, actually, I'm going to go ahead and start sharing the slides for today, and you're going to notice right away that the format is actually quite a bit different than it has been in the past. And I'm hoping that you'll also give me a little bit of feedback um, on these as well. So these are actually lecture notes that are coming from my colleague, Dr. Douglas Tweed, who normally teaches this during the regular school year while I'm off teaching within human biology. And again, this is me. I'm kind of dressed a little bit like my avatar is, and you can always get a hold of me at this email address, and many of you have, so thank you for doing that. And the style of the notes are going to be a little bit different. They're laid out a little bit differently than Dr. Key's notes are, and I hope that either way, that you're going to continue to enjoy all the things that we're going to talk about. And this is actually a really exciting component, so we'll get right into it. So a couple of things to begin with. So for the test and exam, the way that Dr. Tweed had set up his lecture notes, as we go through these lecture series on the CNS, and we'll talk about the CNS, the structure, and everything else today. And then we'll get into sensory systems and the special senses before we wrap up this particular section. The one thing that you are responsible for is that any text that's colored in this lighter gray, you don't need to worry about. Please take down lecture notes as you have been doing. I hope that they've been working out really well for you. Ask questions on discussions. Many of you have. Continue to answer questions for each other. So I've been answering questions as quickly as I can, mainly because I want you to get into the rhythm of studying. But I think at this point, we want to see how things go in tutorials. Ask those questions in tutorials, and maybe I'll wait before trying to take up some of these questions. And if you can, help each other out by also asking. If you need to have some questions in private though, I'm always available, or I may just ask you to post on discussions if I feel like it's more relevant there. Please use my email whenever you feel like it's necessary. And again, a lot of the chapter headings will also be at the very end of the slides, the way that Dr. Tweed had set them out. And I actually really appreciate the comments that were on discussions based on that. So I won't be testing you specifically on any of those materials that's not covered in those lectures, just to be really clear on it. So we're going to go through over the next little while, over the next few lectures on different aspects of the CNS and the nervous system. Today will just be an overview of the central nervous system. Then we'll have an overview or an introduction to the different senses. And then Dr. Tweed has sort of set things up in terms of the optics of the eye itself, the physics of actually seeing, then visual processing, and specializations known as color vision. And then we'll get into some of the other senses, like hearing as well as equilibrium, and then the somatic senses, and then the chemoreceptive senses, things that involve like chemicals involved in smell as well as in taste. It sounds like a really excellent lecture series, and I'm actually looking really forward to bringing you into all of those different things. Now, the other thing that I really love about the way that Dr. Tweed has set this up is that he starts off each of these different lectures with a question of the day, and I've kind of embellished it and added a little bit of my own flavor to this. So the question of the day for him on this section is, are only 4% of the neurons that you're using in your brain right now, sitting there wherever you're listening to this lecture and taking down notes, are only 4% of all of the different neurons, whether it's the 86 billion that's in your brain or the billion that's in your spinal cord, are you only using 4% of your neurons? And again, this gets into you're using only 10% of your brain. You've probably heard that. Where did that number come from? And are you only using this little fraction? And again, great for parties or great for concerts, and if you want to talk about them during concerts. So I'm going to give you a really quick overview of the CNS. We're going to talk about the anatomy, and we've already looked at the energy supply with Dr. Key's notes last week in the modules related to the CSF, and some of that will be a review for you, and it's a great way to review. There's nothing different about these different topics. So the spinal cord will be sort of next as part of the CNS, and then we'll look at the segmental structure of the spinal cord. We'll look at how the spinal cord is organized in terms of the nuclei and tract. And then from that, we'll actually get into what the spinal reflexes actually look like using our knowledge of these nuclei as well as tracts. Then we'll go back into looking at the structure of the brain as well. And then we'll look at the 6 different divisions of the brain, including the brain stem and the cerebrum or the cerebral cortex. Okay, so that sounds like a really exciting outline. So let's just dive right in and go into an overview of the CNS. Okay, so the CNS, or the central nervous system, consists of both the spinal cord down here as well as the brain. And again, you're seeing 2 different views of this, and this is by definition, that's it. This is the spinal cord. And you might be thinking, well, we have nerves elsewhere, so what do those nerves get classified as? And any neurons that we find outside the CNS make up what we call the peripheral nervous system. And this peripheral nervous system, which we abbreviate as the PNS, consists of all of the other neurons and the parts of the neurons that are found outside of the spinal cord and outside of the brain. And generally speaking, this PNS is comprised of the somatic nervous system, so the parts of the nerves that are exiting the spinal cord are out in the periphery outside of the spinal cord that are used for controlling voluntary action via the skeletal muscle cells. And in addition, the PNS will also include the autonomic nervous system that is used to control visceral functions such as heart rate as well as breathing. Now, part of the autonomic nervous system, a sub-branch of the autonomic nervous system, is known as the enteric nervous system. And so this is usually involved in controlling digestive processes, the movement of the gut, and it is getting its input from the spinal cord. But in addition to that, it can also work independently. So think of the spinal cord being able to regulate some of these functions, but in and of itself, the enteric nervous system is capable of just the day-to-day activity of the digestive system as well as movements in the gut. Hoping that makes sense to you. And then going back to the CNS, I mentioned this earlier, the brain has about 86 billion neurons. And again, that's an estimate. No one has actually physically counted every last single neuron in every last single brain, but using a technique known as stereology, neuroscientists have estimated that we have somewhere between 86 billion to about 100 billion neurons, and most neuroscientists now believe it's about 86 billion neurons, and your spinal cord has about 1 billion neurons in terms of the total number there. So again, roughly 87 billion in total, and the estimates for the enteric nervous system range from about 100 to 600 million neurons. And again, the total for the PNS, that other branch that's involved in somatic movement, etc., it's likely going to be similar in terms of numbers. So by far, your brain has the greatest number of neurons that we're looking at. In addition to that, just as a reminder, and we spoke about different types of glial or support cells, and we talked about astrocytes and the importance of astrocytes in the last lecture, and these are used to support as well as protect neurons, and they seem to be just as numerous, and some people say they're more numerous than the neurons that we have. We had already talked about this in the lecture with Dr. Key, but just as a background, so from lecture number 6, the last lecture that you had, we can see these lateral ventricles, and then we have this structure known as the third ventricle, which then connects through the fourth ventricle and then down into this central canal of the spinal cord. And of course, we also saw other structures associated with this, but just sort of as an overview or review of the CSF and the way that it got produced by those specialized ependymal cells that we talked about in the last lecture. So other structures that we need to be able to discuss. When we look at the CNS, the central nervous system, we can actually see a distinction, whether it's on an MRI like this, and this is what a normal MRI would look like, or if we physically examined the CNS, we would actually be able to see differentiation based on color. So we would see that there is gray matter, and this consists of the nerve cell bodies, the somas that we've been looking at, and we've been concentrating on single cell levels, but now we're looking at billions of these at a time. And then the unmyelinated axons, as well as the dendrites. And these cell bodies tend to be arranged in layers, and these are particularly true. We have layers within the brain, or we have them grouped together. Instead of being in layers, we get them grouped together in little balls or ball-like structures, and these are going to be called nuclei. Now, I'm going to show you this a little bit later, and this is near the very end. So around slide 29, 30, I'm going to show you these little clusters of cell bodies within the gray matter, and you'll again be reminded of this. The white matter, though, consists of the myelinated axons running in bundles, which in the brain we call tracts. And we have another name for these tracts that I'll get to in a moment, but generally speaking, we talk about different tracts. Another way that we can call these tracts or bundles of axons that are myelinated is a callosum. And here we see the corpus callosum. And we'll get to what that means in a moment, but this is the white matter down here, and the gray matter is sitting on top here. And literally, it does look very much like that dichotomy that you are seeing here. So in the peripheral system, again, just as a terminology, we are looking at clusters of neurons called ganglia. You'll see that in a few minutes when we say dorsal root ganglia. It means the cell bodies that are sitting outside of the spinal cord. So hopefully that will make sense. And then we call these bundles of axons in the peripheral nervous system, we typically call them nerves. So even though it's a bundle of axons, we still will call them nerves. It's just a terminology that we tend to use. So I spoke to you when I was holding up a pineapple last week, and the pineapple weighs about 3 pounds. 3 pounds is roughly how much your brain weighs, and it's a lot heavier than you would give it credit for. And if the brain is about 3 pounds, an average individual, let's say, is about 150, so comparing male, female, taking an average value of about 150 pounds, that works out to about 2% of the body's mass. And again, you don't need to worry about the math, but we know that we actually have a lot of blood flowing to the brain from the heart. And we also know that astrocytes, from lecture number 6, play a huge role in regulating the blood flow to the brain. So even though it's getting quite a lot of blood flow, about 15%, it actually consumes a greater amount of the total body's glucose. Again, the number here being slightly different, but the key point here between lecture 6 and this lecture number 7, is that the brain consumes a lot more glucose than the amount of blood that's actually flowing through it. And for its size, only 2% of the body's mass, it's actually using a lot more energy than it should be. And when you take a look at it though, on average, you might think, wow, that's a lot of energy for the brain to be expending. But it turns out that if you looked at it in terms of overall energy use, our brain is incredibly efficient. So it runs on the equivalent of about 40 watts, like a light bulb. And again, when you think about the best supercomputer in the world, or up until a few years ago, the supercomputer called Watson, it needs about 200,000 watts to be able to work at the same level as a human brain. So our brains are very, very energy efficient. And that's sort of the main take-home message. And here's that example of grayish colored text that Dr. Tweed likes to use. You don't need to worry about these numbers at all. All right. So the CNS, how does it do this? How does it save this amount of energy? So it saves energy by limiting communication between neurons. So neurons, as you remember, send a lot of action potentials down their axons. And these action potentials take a lot of energy, resetting that membrane potential again. And this is what I'm asking you, do you remember why in the previous week we need those sodium, potassium, ATP pumps functioning to reset? And those ATPases are important. And the energy supply to the CNS can only support a low rate of firing. So in the cortex, it permits an average rate per cell of just 1 spike every 6 seconds. So you might be wondering, wow, that's an awfully low number. I thought there were lots and lots of action potentials going on. And so that means that on average, Remember hearing that we only use like 10% of our brain? This is where that number comes from. But it actually is lower. It's about 4% of your neurons are firing at any given time, even when you're concentrating on a task. And that's because the CNS likes to conserve as much energy as it can, and it supports very, very low rates of firing. Now, what neurons it decides to use at any given time, that small 4%, that's how it actually makes up for low rates of firing. And this is because that communication, it uses up a lot of energy. And so it has to be able, your brain has to be able to use it sparingly. And again, that's one of the things that makes neuroscience so incredibly exciting and so incredibly interesting to study. And I'm hoping that you're starting to pick up on that. So let's move on to the other structure, and we're going to have to talk about the way that it's segmented, the way that it's sort of divided, and how we look at the anatomy of the spinal cord. And I'll also point out a few things that you probably want to spend a little bit more time on when you go back and review your notes, and as you're jotting down these notes. By the way, I'd like to know, do you stop and pause the lectures and then jot down notes? Or do you listen to the whole thing all at once and then come back and review anything that you might have missed? Am I speaking too quickly? Or would you rather that we try something different? Again, I'd really love to hear that type of feedback. And that's something you can email to me directly, as several of you already have. So this is a really busy slide, but I want to take you through this. This is very important to kind of organize you and give you a review of the anatomy and the different terms that we use in terms of the neuroanatomy within the brain. So we have these different spinal cord segments. In fact, we have 31 spinal cord segments that we'll get to in a moment. And if we look at one segment, and this represents one segment of the spinal cord, and each of them have a pair of spinal nerves. There's one on the left and there's one on the right. So that's that pair. And each spinal nerve in turn, so this is one spinal nerve and here is the other, has both a dorsal root, and remember I told you about the ganglia, and this means that the dorsal root ganglion, this larger structure, actually has the cell bodies for the nerve fibers themselves. So each spinal nerve, again, remember nerves are bundles of axons or bundles of neurons, not just one. Each spinal nerve or bundle has a dorsal root up here at the top, and this is carrying information into the spinal cord. So it's coming from the periphery. Action potentials are traveling down this way and entering the spinal cord here. And then in addition, we also have the ventral root, which is carrying efferent information or outgoing signals. So signals are leaving the spinal cord through this ventral root. And often these are motor signals or signals that will travel from the ventral root toward the muscles, skeletal muscles, to cause them to contract. So every time you are picking up that cup, a signal has left a segment of your spinal cord, has traveled down this ventral root and activated specific types of muscles to allow you to pick up that cup. Again, really cool if you think about that. Now, what do all of these names mean? So again, going back to hopefully either your grade 10 or 11 biology, and it might have been a while, or in your anatomy courses, dorsal means toward the back, so dorsal fin on a dolphin, it's on the back of the dolphin, and ventral meaning toward the belly. So this is, again, is oriented, ventral root closer toward the belly side in us as well. So when we look at the gray matter and we look at the white matter, it's the same as we would have within the brain because it's still part of the CNS, and we do have gray matter and we do have white matter, but they're oriented in slightly different ways. And just again, to keep things in hand, this is the gray matter here within the spinal cord, this butterfly shape. And if we look at the different segments of the spinal cord, we will actually see within this butterfly shape, both on the right and on the left, because they're symmetrical and they're paired, we would have what is known as this dorsal horn, meaning that it is projecting toward the dorsal surface, toward the back. And we have a ventral horn, which means, again, projecting toward the belly side. And we have these outcroppings this way. Now, why is that important? And this, again, is a slide, and these slides are important coming up. for you to understand a couple of things that are organized. Remember in the brain, as I showed you a few slides back, that we have these layers of neurons. And it's the same, because we're still talking about the CNS, it's the same within the spinal cord as well. And if we were to look at the functional and anatomical structures and compartments that exist within the spinal cord, we would actually see that within the ventral horn of the dorsal horn of the spinal cord, we would actually have the somatosensory nuclei. And that means that these nuclei in the dorsal horn are receiving sensory signals that come from places like the skin. So just under those, we would have the visceral sensory nuclei. And again, there's sensory signals coming in. So in the dorsal horn, it's always sensory signal, whether it's from the skin or from the internal organs. These are sensory signals that are coming in. And again, dorsal root coming in, dorsal ganglia are where the cell bodies are, and we often see them denoted like that. So information is coming in this way. On the opposite side, we also will have this structure in the ventral horn, and this represents the somatic motor nuclei. And again, these are efferent nuclei. So that means that information from these neurons are leaving the CNS, and these often represent motor nuclei that are sending commands to skeletal muscle. We also will have here the autonomic efferent nuclei, and these are also considered to be on the ventral horn, but they're known to be a little bit more lateral, so that means toward the side, than the ones that are found within the somatic motor nuclei. So these are much more on the ventral surface. These are more lateral or on the side of the ventral horn. Hoping that that makes sense for all of you. And as we go on, we'll also see that if we now are moving away from the gray, that was the previous slide, and now we get into the white matter, this is where we'll actually find a number of different white matter tracks. Again, white matter meaning myelinated tracks, and we would see that there are groups of axons or tracks, and remember within the CNS we call groups of axons tracks. We have ascending tracks in green, localized here, that are carrying signals to the brain, and it's ascending because it's going from a lower structure, the spinal cord, to a higher structure like the brain. These are mainly found on the dorsal surface. So now that we know that this is dorsal, we know that these are sensory signals are coming into the dorsal horn, and then the next neurons are actually carrying those signals up toward the brain using these spaces within the spinal cord. On the other hand, we also have descending tracks, and these are actually carrying information from the brain down to specific levels of the spinal cord, and they may be coming down on to the spinal cord on these blue groups of the spinal cord here. And these are mainly, although not 100%, mainly ventral, which is why it says that on this slide. We'll see a few tracks over here on the lateral aspect as well. And propriospinal tracks that we'll talk about later that give information about how the joints are moving in space actually stay within the spinal cord. So they're actually part of this central gray. And we'll talk about that a little bit later on. So let's put some of this information together, because again, all of this gets to be really interesting, but it can also be challenging unless we actually start integrating this material. So let's look at what we call a spinal reflex. And that means all of the signals and all of the output, so all of the afferent, starting with the letter A, and all of the efferent signals stay within the spinal cord, and we don't actually have to use any of those ascending nor those descending tracks. Okay, so hopefully that makes sense. It's a spinal reflex. Everything is done at the level of the spinal cord. An example of this is that very stereotypic knee-jerk reflex. So a physician who is examining your reflexes will take a little rubber hammer, he'll tap your knee as you're sitting down, and that hammer will stretch a ligament that is just below the kneecap. And as the ligament stretches, it actually will send sensory information into the spinal cord, and then will activate a group of neurons, and this information is coming into the dorsal horn of the spinal cord, and these will then directly cause the innervation of neurons in the ventral horn. So that's why you don't have to think about it. It's done automatically. It's a reflex. Although there are fibers that send branches up to the brain, all of this is done at the level of the spinal cord, and that's why it's a spinal cord reflex. Okay, and that means that it sends out signals through the efferent pathway, that motor output, in order to cause these leg muscles to contract. And again, that can allow for the kicking motion that happens when the physician's rubber hammer strikes that ligament. All right, so let's get into the brain, and let's talk a little bit of some of the interesting things that happen up in the brain as well. So just like we were looking at the spinal cord, there is some structure that's associated with the brain as well. Now, this one is a little bit complex, I guess, if you want to think of it that way. So here we're looking at a sagittal section of the brain, so sideways. And we are looking at the front of the brain here. This is the back of the brain with the cerebellum. So your nose would be here, your eyes would be up here, the back of the head would be back here, just to orient you a little bit. And all of this structure up here, the really colorful structure, is known as the cerebrum, or the cerebral cortex. And the old name for this is the telencephalon, from the embryological origins of the cerebrum. And then the next structure down, which is known as the diencephalon, and this diencephalon consisting of many structures, but the important structures being things like the thalamus. And again, you don't need to write those notes down right now. We'll go into each of these sections in turn. The midbrain, which is just found under that, and this color here, this would be the midbrain, or the mesencephalon. Many of the different types of nuclei that help to fine-tune movement are found within the mesencephalon, or the midbrain. And then we start getting into the brainstem proper, which consists of the pons and the medulla, and these structures here become very important, and we'll talk about them as well. And then we have this other structure here at the back called the cerebellum. All right, so let's keep going. And cerebellum, I'll actually stick up a slide in the next lecture, but if you ever watched the Powerpuff Girls, from a cartoon from a number of years ago, there was a character named Cerebellum, and actually her hair is reminiscent of the cerebellum. Now, I'll put that picture up for you at the beginning of the next lecture, but it's an interesting point that the people and the artists that drew the Powerpuff Girls actually had a sense of humor, and they had a pretty good knowledge of the nervous system as well. So the medulla, the pons, and the hindbrain make up this brainstem that we're looking at here. So again, the thalamus, which is not part of the brainstem, this is part of the diencephalon. We have that mesencephalon here, the pons, and then the medulla. This would all make up the brainstem. And what's important about these structures is that within the brainstem, all three of these, we have a lot of control centers that are really important for autonomic functions that are vital for us to be able to survive. Things like breathing. There are groups of neurons there that control breathing rates. Swallowing, the neurons that will help to control the swallowing reflex. Vomiting, regulating blood pressure, all of these brainstem structures have various nuclei that are involved there. And in addition, we also have these cranial nerves, and we have a total of 12 cranial nerves, and cranial nerves 3 to 10, 10 being the vagus nerve, and the hypoglossal nerve, cranial nerve number 12. Those names are not important, but these cranial nerves all arise from the brainstem. So that means that most of the cranial nerves actually arise from the brainstem. You can actually see some of these cranial nerves having their origins here. Again, the exact ones you don't need to worry about. This is not an anatomy class, but a physiology class. Again, just to give you a different view of it. So here are cranial nerves number 3 all the way down to number 12. The vast majority of them are coming out at the level of the brainstem. So here's the brainstem, the medulla. The pons being over here. So you can see lots of these little cranial nerves that you see jutting out actually come from the brainstem. And we have very few that are originating at the level of the brain itself. Things like the olfactory bulb, again, the name's not being important, cranial nerve number 1, and then the optic nerve, cranial nerve number 2, etc. Whether or not we actually have a cranial nerve 0, anatomically we do, but if you're wondering what this terminal nerve or cranial nerve 0 does, we don't really know. And in fact, it wasn't really found until like about a decade ago, and it was thought to be completely vestigial, something as a remnant of something that happened in evolution. Other organisms have cranial nerve number 0, and they use it to detect pheromones. But as far as we can determine, humans can't detect pheromones. And so we don't really know what cranial nerve 0 is doing, but we do know the origins for most of the cranial nerves, how they exit the CNS. Most of them are at the level of the spinal cord, and a few leave through the brain, okay? And that's sort of the main take-home message from those 2 previous slides. So here we're looking at the diencephalon. So this diencephalon is delineated by just being under this structure known as this corpus callosum. And I mentioned corpus callosum in passing, so it's a tract. I'll get to this tract again. It's a very important defining group of axons within the brain. And it's just under this corpus callosum. And here are the different structures in the diencephalon. So the thalamus, the hypothalamus, which means the thalamus under the thalamus, so the hypothalamus, the pineal gland, and the pituitary gland. Now, do you remember from lecture number 6, when we were talking about the blood-brain barrier, what makes some of these structures unique? Do you remember that at all? Is the blood-brain barrier intact within this area and within the hypothalamus? You might want to go back and revisit those notes, or let me know if you think that the blood-brain barrier might be a little different in these areas, and what other areas do you think the blood-brain barrier is also maybe a little bit different as well. So the thalamus is important. We've already mentioned the thalamus a little bit in different series of slides, and we'll see this again throughout the sensory system. The thalamus is important for processing information as it goes to and leaves the cerebral cortex. So it's a station, it's a hub for information to pass through. The hypothalamus, which is just under this, in sort of this yellowish color here, is important for regulating different behavioral drives. Behavioral drives meaning like eating, when to do things related to eating, or if you're worried and scared, those types of behavioral drives are regulated through the hypothalamus. And they also have a number of endocrine functions and autonomic functions, and the hypothalamus's job is to keep you alive and functioning well. And in addition, the pituitary and pineal glands that we'll run into a little bit later, or if you're taking PSL 301, you'll see the pituitary gland in that section as well for different types of hormones. So the cerebrum has a number of different connecting fibers, and there's some structure associated with it. The most important one that is highlighted for you here between the left and right hemisphere. So here's the left hemisphere, here's the right hemisphere. I mentioned to you that we will come back to basal ganglia. And these basal ganglia, again, ganglia being groups of neurons, and instead of being in this sort of layer structure, which we have here in the cortex in the gray, and here's that white matter, that corpus callosum is a bundle of white matter tracts that will connect the left and right hemisphere. So information travels both left and right through this corpus callosum. And then we have embedded these basal ganglia, which are groups of neurons that are very important in helping to control movement. And again, we will also see, as part of this system, the limbic system as well in a later group of slides. So again, if we look at the two hemispheres, there's this concept known as cerebral lateralization, and that means that some of the different behaviors and cognitive functions, like how we think, how we do different types of behaviors, tend to be found more on one side than on the other. Now, it doesn't mean that it's completely on one side, but we do tend to see favored behaviors and groups of neurons that are responsible for those favored behaviors more on one side than on the other. So things like your abilities for language and math tend to be more on the left hemisphere. Writing tends to be a little bit more localized to the left hemisphere than the right. Speech centers tend to be a little bit more on the left. Again, analysis by touch seems to be more on the right, and spatial analysis and how things look in space, whether you're trying to park a car, etc. That's spatial analysis. The groups of neurons on the right tend to be much more organized. Now, connecting those two left and right hemispheres that seem to have this lateralization, this is that corpus callosum. So information does indeed flow back and forth, and that's important to consider as well. I mean, you can't really do spatial analysis and do math separately. You need that information together, and you can't really do analysis by touch and writing and keep those separately either, nor can you have speech without being able to do a lot of the spatial analysis. You're reading this text and trying to convert it to speech in your head. You need to have communication, and it's not a perfect cerebral lateralization. It just means that we have areas that seem to be more focused on those on one side versus the other. Now, going to each of these hemispheres, so this would be the left hemisphere, we can further define some of the different areas within the left hemisphere, and we call these lobes, and these are separated by these anatomical structures, which we call sulci or fissures, and we'll get to those a little bit later, but this would be the frontal lobe. All of the neurons here would be representative of the frontal lobe. This structure here is a sulcus. We'll get to that later. And everything behind that would be the parietal lobe. There is actually a calcarine fissure that separates the parietal from the occipital. That detail is not important, but there is a functional divide, just like Yonge Street separates Toronto from central Toronto to the east side of Toronto. We have the occipital lobe sort of on after that fissure as well, and this would again be at the very back of your brain. And then the temporal lobe, you're on the sides of your head, we would have these temporal lobes. And again, all of these have different functions, they have specializations, and they're responsible for different things in terms of function and not just anatomy. And we'll get to those a little bit later in the course as well. Now, if that was sort of the outside of the brain, now if we looked at the more along the midline of the brain, we would actually see this unique structure, which is known as the cingulate gyrus. And this cingulate gyrus also helps to define a little bit of what is known as the limbic system. Now, this is sort of more toward the midline. I'll show you that on the next diagram. And this is sort of part of that limbic system. So here's that cingulate gyrus again, part of that limbic system. And this is evolutionarily a very old area of the brain. And there are groups of neurons that seem to be very important. And again, this is sort of the left hemisphere, right hemisphere. Instead of looking at the surface, we're more in the midline, so right near sort of the left-right divide. And if we take a look, the cingulate gyrus is important on both sides. And in addition, there are other structures associated with the limbic system, like the hippocampus, as well as the amygdala. All of these together are known as the limbic system. Now, why is this limbic system important? So this limbic system, including the hippocampus, the amygdala, the cingulate gyrus, etc., all of these are concerned with different types of emotional responses, things like motivation, things like fear, things related to memory, particularly through the hippocampus. And it turns out that with the amygdala, if you induce specific lesions within monkeys and you eliminate the amygdala so that they don't function, and that group of neurons that comprise the amygdala, if you remove them surgically, they are no longer going to be afraid of snakes as much. And in humans, there's actually a genetic condition which is known as Urbach-Wiethe disease. And here, this is a lipid, a lipoproteolytic disorder that actually causes calcification of certain groups of neurons, and you spontaneously will have those groups of neurons dying. And there was a patient who is still alive, so we can actually identify her. She is known as SM. Her initials are SM, and she is patient number 46. She doesn't have functioning amygdala, and she doesn't exhibit the same fear responses, so she's actually really happy when different types of scary responses might be going on. So she would actually not feel so bad going into an exam where she hasn't studied. And maybe that's a good thing, maybe that's a bad thing, I don't know, like you'll have to tell me. But it's a very interesting phenomenon that we can actually see through genetic causes, a lesioning of groups of neurons. Now I'm leaving this one up here. This is sort of a practice question for you. You should be able to answer it either now or after you've gone back and reviewed, but I want you to slowly get ready for the different assignments that are coming your way. I know many of you want to get a head start, so I want to help you as much as you can. So please continue to give me that feedback. And Dr. Tweed has been kind enough in his regular lectures to provide the readings that are associated in Silverthorne's Human Physiology textbook. So if you want more information or you want something to accompany these lecture slides, these are the ones that he had recommended for you. And again, it's only if you want some supplemental information. So I'm going to stop sharing the slides right now. I hope that everything went okay for you. Thank you for the feedback so far. Keep going. There's a lot in this course and you have a lot to do. There's a lot to kind of memorize and to become familiar with. There's more on the way. And so again, I'm hoping you're getting into a rhythm of studying, finding different days and times that work best for you, whether you're doing them in bunches or one at a time. I want you to really remember the most important thing is that you're taking care of yourself and not just studying. And again, we tend to really focus on excellence here at the university, and that's important, and I know it's important to you, but so is your health and so is your well-being. So make sure that you're taking care of those. If you have the opportunity to get outside at any point in time, please do so. And one way to help yourself is that there's a lot of material, I know, but make sure that you're doing it on a consistent level and not just sort of leaving it. This course tends to pile up really quickly, and I think that you're already getting the sense of that. So thank you so much. We're gonna continue to see each other for a few more lectures. Let me know, do you like the way that Dr. Tweed sets his lectures up? Would you rather have the style that Dr. Key was using, basically following their guidance on this and using their notes? But let me know. Is there more that you'd like to know or more practical applications? Do you want more practice questions? Let me know as soon as you can. Smash that like, oh, sorry, wrong thing. It's going to tell you to smash that like button. But I'll see you in the next lecture. All right, take care, everyone.